All right, everyone, let's dive into biological classification. Now, this is one of those chapters that connects everything in biology, so pay close attention. Think of classification as organizing life itself. It's how we make sense of millions of organisms on Earth. Now, since the very beginning of human civilization, people have been classifying living things. But back then, it wasn't scientific at all. People grouped organisms based on what they could use them for. Is this plant good for food? Can I use this animal for clothing? Is this tree useful for shelter? It was all about practical needs, not science. So who was the first person to actually try doing this scientifically? Aristotle. He's the earliest scientist we know who attempted a more scientific basis for classification. What did Aristotle do? He looked at plants and said, okay, I can divide these into 3 categories based on their physical structure: trees, shrubs, and herbs. Pretty simple, right? For animals, he came up with this interesting system. He divided them into 2 groups, those which had red blood and those that didn't. Now we know this isn't the best way to classify, but remember, this was over 2,000 years ago, and it was revolutionary for its time. Fast forward to Linnaeus' time. He developed what we call the 2-kingdom system. Everything was either Plantae, that's all plants, or Animalia, that's all animals. Sounds simple enough, right? But here's the problem. This system was way too simple. It didn't distinguish between prokaryotes and eukaryotes. Think about that. Bacteria and pine trees were both in the same kingdom just because they had cell walls. It lumped together unicellular organisms with multicellular ones. And get this, it put photosynthetic green algae and non-photosynthetic fungi in the same kingdom. That's like putting a solar panel and a vacuum cleaner in the same category, just because they're both household items. So what happened? Scientists realized that a huge number of organisms simply didn't fit neatly into either plants or animals. They needed a better system, one that looked at multiple characteristics: cell structure, how the organism's body is organized, mode of nutrition, reproduction methods, and most importantly, evolutionary relationships. This is where classification started becoming truly scientific. Before we move to the modern system, let me show you a question that appeared in NEET 2023 about nomenclature, which Linnaeus actually gave us. Here's the question: X and Y are the components of binomial nomenclature, and this naming system was proposed by Z. The correct answer is X is the generic name, that's the genus. Y is the specific epithet, that's the species name. And Z is Carolus Linnaeus. Remember this. Every organism gets 2 names, genus and species. That's binomial nomenclature, and Linnaeus gave us this system that we still use today. Even though his classification had limitations, his naming system was brilliant, and we still use it. So while we're talking about better classification systems, we should remember that Linnaeus contributed something invaluable that lasted. All right, now we come to the system you really need to know for your exam: Whittaker's 5-kingdom classification, proposed in 1969 by our Whittaker. This is the framework for this entire chapter. He divided all living organisms into 5 kingdoms. Let me give you a quick overview. Kingdom Monera includes all bacteria. Kingdom Protista has all the single-celled eukaryotes. Kingdom Fungi includes organisms like mushrooms and yeasts. They're heterotrophic and have chitin in their cell walls. Kingdom Plantae includes all chlorophyll-containing organisms that photosynthesize. And Kingdom Animalia includes all multicellular heterotrophic organisms. Now, each of these kingdoms has very specific characteristics that set them apart. What criteria did Whittaker use to create these 5 kingdoms? This is important because exams love testing this. First, cell structure. Is it prokaryotic or eukaryotic? Second, body organization. Does it go from just cellular level to tissue level to organ system? Third, mode of nutrition. Autotrophic or heterotrophic? Fourth, reproduction methods. How does it reproduce? And fifth, phylogenetic relationships. How are organisms evolutionarily related? These 5 criteria together gave us a much more scientific and comprehensive classification system. Now your textbook has this incredibly important table, Table 2.1. that compares all 5 kingdoms across different characteristics. You need to know this table inside out. It shows cell type, whether prokaryotic or eukaryotic, cell wall, whether present or absent, and if present, what it's made of, nuclear membrane, present or absent, body organization, from cellular to organ system level, and mode of nutrition, the different nutritional strategies. This table is your best friend for understanding how these kingdoms differ from each other. I highly recommend you make a chart and memorize these distinctions because questions are directly asked from this comparison. Now, science doesn't stand still. There's a more recent system called the 3-domain system that actually divides kingdom Monera into 2 domains, bacteria and archaea, and puts all the remaining eukaryotic kingdoms into a 3rd domain. This gives us essentially a 6-kingdom classification. You'll study this in detail in higher classes, but it's good to know it exists. The point here is that classification systems are dynamic. They change and evolve as our understanding of organisms improves. We discover new characteristics. We understand evolutionary relationships better through DNA analysis, and the classification system updates accordingly. What seemed correct in Linnaeus' time changed by Whittaker's time, and it's still changing today. That's how science works. It's self-correcting and always improving. Let's start exploring the kingdoms in detail, beginning with kingdom Monera. The sole members of this kingdom are bacteria. Now, when I say bacteria are abundant, I mean really abundant. They are the most abundant microorganisms on Earth. We're talking about organisms that are literally everywhere, and I mean everywhere. Just to give you an idea of how abundant, if you pick up a handful of soil, there are hundreds of bacteria in just that one handful. Hundreds. They live in extreme habitats where very few other life forms can survive. Hot springs with boiling water, deserts with no water, snow and ice in Antarctica, deep oceans under crushing pressure. And many of them live in or on other organisms as parasites. So from the harshest environments to inside your own gut, bacteria are everywhere. Now, bacteria are grouped into 4 categories based on their shape, and you absolutely need to know these. First, coccus. These are spherical or round bacteria, and the plural is cocci. Second, bacillus. Rod-shaped bacteria, plural is bacilli. Third, vibrium. Comma-shaped bacteria, plural is vibrio. And fourth, spirillum. Spiral-shaped bacteria, plural is spirilla. Think of it as spheres, rods, commas, and spirals. Shape is often the first thing we use to identify bacteria under a microscope. Here's what makes bacteria truly remarkable: their metabolic diversity. The chapter says bacteria as a group show the most extensive metabolic diversity compared to many other organisms. What does this mean? Some bacteria are autotrophic. They synthesize their own food from inorganic substrates. And within autotrophs, you've got photosynthetic autotrophs that use light energy and chemosynthetic autotrophs that use chemical energy. Then you've got heterotrophic bacteria, the vast majority that depend on other organisms or dead organic matter for food. This incredible metabolic flexibility is why bacteria can survive literally anywhere. The textbook makes this fascinating point. Though bacterial structure is very simple, they are very complex in behavior. Think about it. They're just single cells, no nucleus, very basic structure. But their behavior, their metabolic capabilities, extremely complex and diverse. This is why bacteria have survived for billions of years and why they'll probably outlive every other organism on Earth. Simple structure, complex function. That's the bacterial strategy for success. Now within bacteria, we have a special group called Archaebacteria. What makes them special? These bacteria live in some of the most harsh habitats on Earth. And the reason they can do this is because they have a different cell wall structure compared to other bacteria. This unique cell wall structure is responsible for their survival in extreme conditions. So it's not magic, it's their cell wall chemistry that protects them. There are 3 main types of Archaebacteria you need to know. First, Halophiles. These love salt. Halo means salt, phile means loving. They live in extremely salty areas like salt lakes and brine pools where the salt concentration would kill most organisms. Second, Thermoacidophiles. Thermo means heat, acido means acid. These guys thrive in hot springs where the water is boiling hot and acidic. Imagine that environment. Most organisms would be cooked and dissolved, but thermoacidophiles are happy there. Third, Methanogens. These are found in marshy areas, and here's something interesting. They're also present in the gut of ruminant animals like cows and buffaloes. So why are methanogens in cow and buffalo guts important? They're responsible for the production of methane biogas from the dung of these animals. This has practical applications. We can collect this biogas and use as fuel. So methanogens aren't just scientifically interesting, they have economic importance too. This is a good example of how understanding microbiology has real-world applications. Let me show you an NEET question about Archaebacteria habitats. Here's a 2019 NEET question. Match the organisms in column 1 with habitats in column 2. Halophiles, Thermoacidophiles, Methanogens, and Cyanobacteria with hot springs, aquatic environment, guts of ruminants, and salty areas. The correct answer is Halophiles match with salty areas, Thermoacidophiles with hot springs, Methanogens with guts of ruminants, and Cyanobacteria with aquatic environment. See how NEET directly tests whether you know which bacteria lives where? And notice they mixed in Cyanobacteria. We'll talk about those next to test if you're confusing Archaebacteria with Eubacteria. By the way, 2017 and 2016 also had questions on this. One asking which bacteria are found in extreme saline conditions with the answer being Archaebacteria, and another asking about primitive prokaryotes producing biogas from ruminant dung, answer being Methanogens. So you can see this topic is a favorite for NEET. Make absolutely sure you know. Halophiles equals salt, Thermoacidophiles equals hot and acidic, Methanogens equals marshy areas and ruminant gut, and all three have special cell walls that let them survive these extreme conditions. Moving on to Eubacteria. These are called the true bacteria. There are thousands of different Eubacteria. They're characterized by the presence of a rigid cell wall, and if they're motile, meaning if they can move, they have a flagellum. Now within Eubacteria, we have several important groups based on their mode of nutrition. First group, Cyanobacteria, also referred to as blue-green algae. Now pay attention, they're called algae, but they're actually bacteria. They have chlorophyll similar to green plants, and they're photosynthetic autotrophs, meaning they make their own food using sunlight. They can be unicellular, single cells, colonial, groups of cells, or filamentous, chains of cells. These colonies are generally surrounded by a gelatinous sheath for protection. Now here's something really important. Some of these organisms can fix atmospheric nitrogen. They do this in specialized cells called heterocysts. The examples you need to remember are nostoc and Animalia. Both have heterocysts and fix nitrogen. This is ecologically crucial because nitrogen fixation converts atmospheric nitrogen into forms that plants can use. Let me show you a 2022 NEET question that tests exactly this concept. The question asked, which of the following is a correct statement? And the correct answer was, Cyanobacteria are a group of autotrophic organisms classified under kingdom Monera. The wrong option said bacteria are exclusively heterotrophic. Wrong, because Cyanobacteria are autotrophic. Another option said slime molds are classified under Monera. Wrong, they're protists. Another said mycoplasma have cell walls. Wrong, they don't. See how knowing that Cyanobacteria are autotrophic bacteria is directly tested? Make sure this is crystal clear in your mind. Second group of Eubacteria, chemosynthetic autotrophic bacteria. These are fascinating. They oxidize various inorganic substances such as nitrates, nitrites, and ammonia. When they oxidize these substances, energy is released, and they use this released energy for their ADP production. So instead of using sunlight like photosynthetic bacteria, they use chemical energy. Now here's why they're important ecologically. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Without these bacteria, these nutrients would get locked up in forms that other organisms can't use. They're essential for maintaining nutrient cycles in ecosystems. Third group, Heterotrophic bacteria. These are the most abundant in nature. The majority of them are important decomposers. They break down dead organic matter and release nutrients back into the environment. But heterotrophic bacteria aren't just decomposers. Many of them have significant positive impact on human affairs. They're helpful in making curd from milk. You've probably eaten yogurt or curd, right? That's bacteria doing their job. They're used in production of antibiotics, life-saving medicines. Some fix nitrogen in legume roots. Plants like peas and beans have bacteria in their root nodules that fix nitrogen for the plant. So heterotrophic bacteria can be incredibly beneficial. But here's the flip side. Some heterotrophic bacteria are pathogens causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker. These are all well-known diseases caused by different bacteria. So bacteria can be heroes or villains depending on the species. This is why studying bacteria is so important. We need to encourage the beneficial ones and control the pathogenic ones. The same group of organisms giving us antibiotics can also give us deadly diseases. Now we come to a very special type of bacteria, mycoplasma. What makes mycoplasma unique? They completely lack a cell wall. Think about that. All other bacteria have cell walls, but mycoplasma don't. They're the smallest living cells known, the tiniest living things we've discovered. And they can survive without oxygen. They're anaerobic. Many mycoplasma are pathogenic in animals and plants, causing various diseases. Let me show you a 2022 NEET question about mycoplasma. It gave two statements. Statement 1, mycoplasma can pass through less than 1 micron filter size. Statement 2, mycoplasma are bacteria with cell wall. Which statements are correct? The answer is, Statement 1 is correct, but Statement 2 is incorrect. Mycoplasma can pass through very fine filters because they're so small. That's true. But they do not have cell walls. That's false. This is a classic example of how NEET tests your precise knowledge. You need to know both facts. Mycoplasma are the smallest living cells, and they lack cell walls. 2017, 2016, and 2015 also had questions on this. One asked, which are the smallest living cells known without a definite cell wall, pathogenic to plants and animals, and can survive without oxygen? Answer, mycoplasma. Another asked, cell wall is absent in which organism? Answer, mycoplasma. See the pattern? NEET repeatedly tests that mycoplasma lack cell walls. This makes them resistant to antibiotics that target cell wall synthesis. That's clinically important. So remember, smallest living cells, no cell wall, anaerobic, pathogenic. Now let's talk about how bacteria reproduce. Mainly, bacteria reproduce by fission, specifically binary fission. The cell simply divides into two identical daughter cells. It's quick, it's efficient, and it's why bacterial populations can grow so rapidly. Under unfavorable conditions, bacteria can produce spores. These spores are extremely resistant and can survive harsh conditions. When conditions improve, the spore germinates and becomes an active bacterial cell again. Bacteria also have a sort of sexual reproduction, though it's very primitive. They can adopt a primitive type of DNA transfer from one bacterium to another. This is important because it allows genetic variation and is one way bacteria can acquire antibiotic resistance. They can literally pass resistance genes to each other. This genetic exchange is one reason why antibiotic resistance is such a huge problem in medicine today. Here's a 2017 question. DNA replication in bacteria occurs when? The answer is prior to fission. This makes sense, right? Before a bacterium divides into two cells, it needs to replicate its DNA so each daughter cell gets a complete copy. So the sequence is DNA replication first, then cell division. Remember, bacteria don't have a formal S phase like eukaryotes, but they do replicate DNA before dividing. All right, moving to kingdom Protista. This is where things get interesting and a bit fuzzy. All single-celled eukaryotes are placed under Protista. But here's the thing. The boundaries of this kingdom are not well defined. The textbook actually says what may be a photosynthetic protistan to one biologist may be a plant to another. So there's some debate and overlap here, which tells you that classification isn't always black and white. It can be subjective depending on which characteristics you emphasize. In this textbook, we include 5 major groups under Protista: chrysophytes, dinoflagellates, euglonoids, slime molds, and protozoans. Each of these groups is quite different from the others, which shows you the diversity within this kingdom. It's almost like a catch-all category for single-celled eukaryotes that don't fit neatly into fungi, plants, or animals. Members of Protista are primarily aquatic. Most live in water environments. This kingdom forms a link with the others dealing with plants, animals, and fungi. Think of protists as the connecting bridge between simple prokaryotes and complex multicellular organisms. Being eukaryotes, the protistan cell body contains a well-defined nucleus and other membrane-bound organelles. This is what separates them from bacteria. Some have flagella or cilia for movement, which we'll see in specific examples. Protists can reproduce both asexually and sexually. Sexual reproduction in protists involves a process of cell fusion and zygote formation. This is more complex than bacterial reproduction and shows the evolutionary advancement of eukaryotes. Now let's look at each group in detail. Before we dive into details, here's a 2016 NEET question. Chrysophytes, euglonoids, dinoflagellates, and slime molds are included in which kingdom? The answer is Kingdom Protista. This is a straightforward question testing if you know which organisms belong to this kingdom. Make sure you can list all 5 groups that we're including under Protista. First group under Protista, chrysophytes. This group includes diatoms and golden algae, also called dimids. They're found in both freshwater and marine environments, so they're pretty widespread. These organisms are microscopic and float passively in water currents. We call such organisms plankton. Most of them are photosynthetic, meaning they make their own food using sunlight, so they're autotrophic. Now, diatoms have this really unique cell wall structure. The cell walls form 2 thin, overlapping shells, which fit together as in a soapbox. You know how a soapbox has a lid that fits over the base? That's exactly how diatom shells fit together. And here's what's special. The walls are embedded with silica. Silica is basically glass, so these walls are indestructible. Even after the diatom dies, the cell wall remains intact. Because diatom walls are indestructible, they accumulate over billions of years. This accumulation is referred to as diatomaceous earth. These deposits can be huge. We're talking about billions of years of diatom cell walls piling up. Now, diatomaceous earth has a gritty texture because of the silica, and we use it commercially in polishing and in filtration of oils and syrups. So these tiny organisms that lived millions of years ago are economically useful to us today. But here's the most important thing about diatoms ecologically. They are the chief producers in the oceans. When we talk about ocean food chains, diatoms are at the foundation. They photosynthesize and create the organic matter that supports all marine life. Without diatoms, the entire ocean ecosystem would collapse. They're that important. NEET loves asking about diatoms. 2018 asked, which organisms are known as chief producers in the oceans? Answer: diatoms. 2016 asked, in which group do cell walls form 2 thin, overlapping shells? Answer: chrysophytes. Another 2016 question had a wrong statement saying walls of diatoms are easily destructible. That's false. They're indestructible because of silica. So remember, overlapping shells like soapbox, silica embedded, making them indestructible, accumulate as diatomaceous earth, and most importantly, chief producers in oceans. Second group, dinoflagellates. These organisms are mostly marine and photosynthetic. What's interesting is they appear in different colors: yellow, green, brown, blue, or red, depending on the main pigments present in their cells. So if you're looking at ocean water and see different colors, it might be different dinoflagellates with different pigment combinations. Dinoflagellates have a cell wall with stiff cellulose plates on the outer surface. These plates give them a somewhat armored appearance. Most of them have two flagella, and their arrangement is unique. One flagellum lies longitudinally, running lengthwise, and the other lies transversely in a furrow between the wall plates, running across. This arrangement of flagella helps them move in a spinning motion, which is actually how they got their name. Dino means spinning. Now here's a phenomenon you might have heard about: red tides. Very often, red dinoflagellates, for example Iyal.App, undergo such rapid multiplication that they make the sea appear red. The whole ocean or large patches of it turn red because of billions and billions of these red dinoflagellates multiplying. But red tides aren't just a visual phenomenon. Toxins released by such large numbers of dinoflagellates may even kill other marine animals such as fishes. So red tides can be devastating for marine ecosystems and even for coastal economies that depend on fishing. Remember Iyal.App as the example that causes red tides. Third group, euglenoids. These are really fascinating organisms. The majority of them are freshwater organisms found in stagnant water, like ponds that don't have much flow. Now, unlike most other organisms, euglenoids don't have a cell wall. Instead, they have a protein-rich layer called pellicle. This pellicle makes their body flexible. They can bend and change shape, which is something organisms with rigid cell walls can't do. This flexibility helps them move through water more efficiently. Euglenoids have two flagella, one short and one long. The longer flagellum is primarily used for movement, pulling the organism through water, while the shorter one helps with steering and sensing the environment. Here's what makes euglenoids truly special: their nutritional flexibility. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Think about that. They can switch between plant-like and animal-like nutrition depending on conditions. This ability is called mixotrophy. In sunlight, they act like plants. In darkness, they act like animals. How cool is that? This makes them incredibly adaptable. And here's something the textbook specifically says is interesting. The pigments of euglenoids are identical to those present in higher plants. This suggests an evolutionary connection between euglenoids and plants. They might represent an intermediate evolutionary stage. The main example you need to remember is Euglena. That's the most commonly cited euglenoid. Now let me show you a 2024 NEET question that tested exactly this flexibility. The question asked you to match organisms with modes of nutrition. For euglenoid, the correct answer was switching between photosynthetic and heterotrophic mode. For dinoflagellate, it was photosynthetic. For slime mold, it was saprophytic. And for plasmodium, it was parasitic. This question specifically tested whether you understand that euglenoids have this unique ability to switch nutritional modes while others are fixed in one mode. Make sure you remember this. It's a distinguishing feature of euglenoids, and NEET loves testing it. Fourth group, under Protista, slime molds. These are saprophytic protists, meaning they feed on dead organic matter. The way they feed is interesting. The body actually moves along decaying twigs and leaves, engulfing organic material as it goes. It's like a living blob that crawls over dead plant material and absorbs nutrients from it. Under suitable conditions, slime molds form an aggregation called plasmodium. Now be careful. This plasmodium is not the same as Plasmodium, the malarial parasite. It's just a term for the mass formed by slime molds. And this plasmodium can grow and spread over several feet. Several feet. For a single-celled organism, that's enormous. You could see it with your naked eye. During unfavorable conditions, maybe it gets too dry or too cold, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. These spores possess true walls and are extremely resistant. They can survive for many years, even under adverse conditions. When conditions improve, the spores germinate and start a new slime mold. The spores are dispersed by air currents, which is how slime molds spread to new locations. This life cycle, growing as a plasmodium when conditions are good, forming resistant spores when conditions are bad, is a brilliant survival strategy. Last group under Protista, protozoans. Now, all protozoans are heterotrophs. They can't make their own food. They live as either predators or parasites. The textbook says they are believed to be primitive relatives of animals, which makes sense because they're heterotrophic and many of them can move. They're like simplified versions of animals at the single-cell level. There are 4 major groups of protozoans, and you need to know all 4. First, amoeboid protozoans. These organisms live in fresh water, seawater, or moist soil, pretty much anywhere that's wet. They move and capture their prey by putting out pseudopodia, false feet. Pseudo means false, podia means feet. They extend parts of their cell membrane and cytoplasm to move and engulf food. The classic example is amoeba. You've probably seen pictures of it in your junior classes. Marine forms have silica shells on their surface for protection. Some amoeboid protozoans are parasitic, like entamoeba, which causes amoebic dysentery in humans. So they can be free-living or parasitic. Second, flagellated protozoans. The members of this group are either free-living or parasitic, and they have flagella for movement. The parasitic forms cause diseases such as sleeping sickness, which is a serious disease in parts of Africa. The example you need to remember is trypanosoma. This is the organism that causes sleeping sickness. It's transmitted by the tsetse fly and can be fatal if untreated. So flagellated protozoans include both harmless free-living species and dangerous parasites. Third, ciliated protozoans. These are aquatic and actively moving organisms because of the presence of thousands of cilia covering their surface. Cilia are like tiny hairs that beat in coordinated waves to move the organism through water. They have a cavity called a gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be steered into the gullet. It's like they create a current that brings food to their mouth. The example is Paramecium, spelled Premoecium. This is one of the most studied protozoans in biology. A 2018 NEET question asked, ciliates differ from all other protozoans in what way? The answer is having 2 types of nuclei. Ciliates have a macronucleus that controls daily functions and a micronucleus involved in sexual reproduction. This is unique among protozoans. The wrong option said using flagella. No, they use cilia, having contractile vacuole. Many protozoans have this. Using pseudopodia. No, that's amoeboid protozoans. So the distinguishing feature of ciliates is 2 types of nuclei. Fourth, sporozoans. This group includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious member is Plasmodium. This is the malarial parasite that causes malaria. The textbook says malaria has a staggering effect on human population, and that's an understatement. Malaria kills hundreds of thousands of people every year, mostly children in Africa. Plasmodium has a complex life cycle involving both mosquitoes and humans. Understanding its life cycle is crucial for controlling malaria. So when you hear sporozoan, think spore-like stage, mostly parasitic, Plasmodium causes malaria. All right, moving to kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show great diversity in morphology, their physical forms, and in habitat. They have cosmopolitan distribution. That means they're found worldwide in air, water, soil, and on animals and plants. They do prefer to grow in warm and humid places, which is why food spoils faster in warm, humid conditions. That's why we keep food in the refrigerator, to prevent fungal and bacterial growth. You've encountered fungi in many forms. The common mushroom you eat, that's a fungus. Toadstools, those are fungi too, usually poisonous ones. You must have seen white spots on mustard leaves. That's due to a parasitic fungus. Some unicellular fungi like yeast are used to make bread and beer. Yeast fermentation is what makes bread rise and produces alcohol in beer. Other fungi cause diseases. Wheat rust-causing Puccinia is an important example that causes huge agricultural losses. Some fungi are the source of antibiotics. Penicillium is the classic example that gave us penicillin. So fungi can be food, they can cause diseases, they can give us medicines, they're economically very important. Now, with the exception of yeast, which are unicellular, fungi are filamentous. What does that mean? Their bodies consist of long, slender, thread-like structures called hyphae. Singular is hypha, plural is hyphae. The network of hyphae is known as mycelium. So when you see mold growing on bread, that fuzzy growth is actually a mycelium, a network of thousands of hyphae. The cell walls of fungi are composed of chitin and polysaccharides. This is important, not cellulose like plants. Chitin is the same material that makes up insect exoskeletons. This difference in cell wall composition is one reason why fungi are in a separate kingdom from plants. Now, some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called cenocytic hyphae. Cenocytic means many nuclei in a common cytoplasm with no separations. Other hyphae have septae or cross walls. These are septate hyphae. The septa divide the hypha into compartments, but even then, there are usually pores that allow cytoplasm and nuclei to move between compartments. NEET has tested this. A 2016 question asked, which statement is wrong for fungi? The answer was, all fungi possess a purely cellulosic cell wall. That's wrong because fungal cell walls have chitin, not cellulose. A 2015 question asked, cell wall is absent in which organism? Answer, mycoplasma, not fungi. And a 2019 question said, yeasts have filamentous bodies with long, thread-like hyphae. That's wrong. Yeasts are unicellular. So make sure you know, fungal cell walls have chitin. Most fungi are filamentous, but yeasts are the exception, being unicellular. Now, let's talk about how fungi get their nutrition. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates. These are called saprotrophic. They're decomposers, breaking down dead leaves, logs, dead animals, and releasing nutrients back into the soil. Some fungi depend on living plants and animals. These are called parasites. For example, rust fungi on wheat are parasites that attack living wheat plants, causing disease and reducing crop yield. So parasitic fungi cause economic losses. Fungi can also live as symbionts in association with other organisms for mutual benefit. The best example is lichens. That's an association with algae where the fungi provide shelter and absorb water and minerals while the algae photosynthesize and provide food. We'll discuss lichens in detail later, but it's a classic mutualistic relationship. Both partners benefit. Another symbiotic relationship is mycorrhiza. That's an association with roots of higher plants. The fungal hyphae extend into the soil much farther than plant roots can, helping the plant absorb water and minerals, especially phosphorus. In return, the plant provides the fungus with carbohydrates made through photosynthesis. This mycorrhizal association is extremely common. Most trees have mycorrhizae, and they grow much better with these fungal partners than without them. Now here's an important NEET 2024 question. Which one of the following is not a criterion for classification of fungi? The answer is mode of nutrition. Why? Because all fungi are heterotrophic. Whether they're saprotrophic, parasites, or symbionts, they're all heterotrophic. They can't make their own food. So mode of nutrition doesn't help us classify fungi into different groups because it's the same for all. What is used for classification? Morphology of mycelium, whether it's septate or cenocytic, mode of spore formation, sexual or asexual, and fruiting body structure. These vary among fungal groups and are used to classify them. Reproduction in fungi is quite diverse. First, vegetative reproduction can take place by fragmentation. The mycelium breaks into pieces, and each piece grows into a new fungus. Fission. In unicellular yeasts, the cell divides, and budding. Again, in yeasts, a small bud grows from the parent cell. For asexual reproduction, fungi produce spores. These spores can be called different names. Conidia, produced exogenously, meaning outside, on the surface of specialized hyphae. Sporangiospores, produced inside a sac-like structure called sporangium, or zoospores. These are motile, meaning they can swim using flagella. Sexual reproduction in fungi is more complex and happens in 3 steps. Remember this sequence. Step 1. Plasmogamy. This is fusion of protoplasms between 2 motile or non-motile gametes. The cytoplasm of 2 cells fuses together. Step 2. Karyogamy. This is fusion of 2 nuclei. The nuclei from both cells fuse to form a diploid nucleus. Step 3. Meiosis occurs in the zygote, resulting in haploid spores. So the sequence is cells fuse, then nuclei fuse making diploid, then meiosis makes haploid spores again. A 2021 NEET question tested this terminology. It asked which statement is correct, and the answer was fusion of protoplasms between 2 motile or non-motile gametes is called plasmogamy. The wrong option said fusion of 2 cells is called karyogamy. No, that's nuclear fusion, not cell fusion. Another said organisms, depending on living plants, are saprotrophic. No, those are parasites. Another said atmospheric nitrogen is fixed in sheath cells. No, it's heterocysts. So make sure the terminology is clear. Plasmogamy equals cytoplasmic fusion. Karyogamy equals nuclear fusion. Now here's something unique about higher fungi, Ascomycetes and Basidiomycetes. In some fungi, plasmogamy immediately results in diploid cells. But in Ascomycetes and Basidiomycetes, there's an intervening dikaryotic stage. Dikaryotic means N plus N, 2 nuclei per cell, but they don't fuse immediately. Each cell has 2 separate nuclei, 1 from each parent. This condition is called a dikaryon, and the phase is called dikaryophase. Later, the parental nuclei fuse, making the cells diploid. Then meiosis occurs. The sexual spores are produced in distinct structures called fruiting bodies. The morphology of a mycelium, mode of spore formation, and fruiting bodies. These form the basis for dividing fungi into various classes. Let's look at these classes now. First class of fungi, Phycomycetes. The name phyco suggests algae-like characteristics, which is why they're sometimes called algal fungi. Members of Phycomycetes are found in aquatic habitats, on decaying wood in moist and damp places, or as obligate parasites on plants. Obligate means they must be parasites. They can't survive any other way. Their mycelium is a septate, meaning no cross walls, no septa, and cenocytic, meaning it's a continuous tube filled with multinucleated cytoplasm. So picture a long tube with many nuclei floating in the cytoplasm, no compartments. Asexual reproduction takes place by zoospores, which are motile. They can swim, or by aplanospores, which are non-motile. These spores are endogenously produced in sporangium. Endogenous means produced inside. For sexual reproduction, a zygospore is formed by fusion of 2 gametes. These gametes can be isogamous, similar in morphology, so you can't tell male from female, or anisogamous, slightly different, or oogamous, dissimilar, where you can clearly identify a large egg and a small sperm. So Phycomycetes show a range from primitive to more advanced sexual reproduction. The examples you need to remember: Mucor, Rhizopus. This is the bread mold mentioned earlier. You've probably seen it as black fuzzy growth on old bread. And Albugo. This is the parasitic fungus on mustard that causes white rust. These are all Phycomycetes. Let me show you an NEET question about these examples. 2024 NEET asked you to match organisms: Rhizopus, Iyal.App, Puccinia, and Agaricus with characteristics. The correct matches were: Rhizopus is bread mold, Iyal.App is smut fungus, Puccinia is rust fungus, and Agaricus is mushroom. So you need to know not just the scientific names, but also the common names. Rhizopus equals bread mold equals Phycomycete. Remember this for your exam. A 2015 question said which statement is wrong, and the answer was Mucor has biflagellate zoospores. That's incorrect. Mucor produces non-motile aplanospores, not biflagellate zoospores. This is testing whether you know the details of asexual reproduction in phycomycetes. Some produce motile zoospores, but Mucor specifically doesn't. Second class, Ascomycetes, commonly known as sac fungi. Why sac fungi? Because they produce their sexual spores in sac-like structures. Ascomycetes are mostly multicellular, for example penicillium, or rarely unicellular, for example yeast, scientific name Saccharomyces. Their mycelium is branched and septate, so unlike phycomycetes, these have cross walls in their hyphae. They can be saprophytic, feeding on dead matter, decomposers, parasitic, attacking living organisms, or coprophilous. Coprophilous means growing on dung. Copper means dung, phyllis means loving. Some Ascomycetes specifically grow on animal dung. For asexual reproduction, Ascomycetes produce spores called conidia. These are produced exogenously, on the outside, on special mycelium called conidiophores. Conidio means conidium, from means bearer. So conidiophores are conidium-bearing structures. When conidia germinate, they produce mycelium and the cycle continues. 2022 NEET asked, identify the asexual reproductive structure associated with penicillium. The answer is conidia. Not zoospores, not gemmules, not buds, conidia. Penicillium is an ascomycete and produces conidia asexually. This is a straightforward question testing whether you know the spore type for Ascomycetes. For sexual reproduction, they produce spores called ascospores, which are produced endogenously, meaning inside, in sac-like structures called asci. Singular is ascus, plural is asci. These asci are arranged in different types of fruiting bodies called ascocarps. So the characteristic feature of Ascomycetes is producing sexual spores inside a sac, the ascus. Examples of Ascomycetes: Aspergillus, commonly found on store grains. Claviceps produces ergot alkaloids, and Neurospora. Neurospora is used extensively in biochemical and genetic work. It was actually used in experiments that helped establish the one-gene-one enzyme hypothesis. Many members like morels and truffles are edible and are considered delicacies, very expensive foods in restaurants. So Ascomycetes include disease-causing fungi, research organisms, and gourmet foods. 2019 NEET asked which statement is incorrect, and the answer was, morels and truffles are poisonous mushrooms. That's wrong. They're edible delicacies, not poisonous. The correct statements were, claviceps is a source of alkaloids and LSD. Conidia are produced exogenously and ascospores endogenously, and yeasts are useful in fermentation. A 2015 question asked which statement is wrong, and the answer was, morels and truffles are poisonous mushrooms. Again, they're edible. So NEET has repeatedly tested this. Morels and truffles are Ascomycetes, and they're edible, considered delicacies. Third class, Basidiomycetes. Commonly known forms of Basidiomycetes are mushrooms, bracket fungi, and puffballs. These are the fungi you typically see in forests. They grow in soil, on logs, and tree stumps. Those shelf-like fungi you see growing out of dead trees are bracket fungi, which are Basidiomycetes. Some grow in living plant bodies as parasites. For example, rusts and smuts that attack crops. The mycelium is branched and septate, similar to Ascomycetes. Here's something interesting about Basidiomycetes. Asexual spores are generally not found. They mostly reproduce vegetatively by fragmentation. The mycelium breaks apart, and each fragment grows into a new fungus. For sexual reproduction, sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. So they don't have specialized sex cells. Regular cells from two different mating types fuse. The resultant structure after plasmogamy is dikaryotic, 2 nuclei per cell. This ultimately gives rise to a structure called basidium, plural basidia. In the basidium, karyogamy happens, the nuclei fuse, and then meiosis takes place, producing 4 basidiospores. These basidiospores are exogenously produced on the basidium. They form on the outside surface. The basidia are arranged in fruiting bodies called basidiocarps. When you eat a mushroom, you're eating the basidiocarp, the fruiting body. The actual fungus is the mycelium underground or inside the wood. Common examples. Agaricus. This is the common edible mushroom you buy in stores. Eustalago. This is smut fungus that attacks corn and other crops. Puccinia. This is rust fungus that attacks wheat and causes huge agricultural losses. We mentioned Puccinia earlier when talking about wheat rust. So basidiomycetes include edible mushrooms and serious crop pathogens. Fourth class, deuteromycetes, commonly known as imperfect fungi. Why imperfect? Because only the asexual or vegetative phases of these fungi are known. The sexual stage either hasn't been discovered or doesn't exist. So they're called imperfect because the life cycle is incomplete. We only know the asexual part. Once you discover the sexual stage, the fungus is considered perfect and can be classified properly. When the sexual forms of these fungi are discovered, they are moved into classes they rightly belong to, usually Ascomycetes or Basidiomycetes. Here's where it gets confusing. It's possible that the asexual and vegetative stage have been given one name and placed under deuteromycetes, and the sexual stage another name and placed under another class. Later, when scientists established the linkages, when they realized these are the same fungus at different stages, the fungus was correctly identified and moved out of deuteromycetes. So deuteromycetes is kind of a temporary holding category for fungi whose full life cycle we haven't figured out yet. 2015 NEET asked, the imperfect fungi, which are decomposers of litter and help in mineral cycling, belong to which class? Answer, deuteromycetes. This is testing whether you know what deuteromycetes do ecologically. Many of them are decomposers breaking down plant litter and helping recycle minerals in ecosystems. Deuteromycetes reproduce only by asexual spores known as conidia. No sexual reproduction. That's why they're imperfect. The mycelium is septate and branched, similar to Ascomycetes and Basidiomycetes. Ecologically, some deuteromycetes are saprotrophic or parasites, but a large number of them are decomposers of litter, helping in mineral cycling. They break down dead plant material and release nutrients. Examples you should know. Alternaria causes leaf spot diseases on many plants. Calototrichum causes anthracnose diseases, and trichoderma. This one is actually beneficial, used as a biological control agent against other pathogenic fungi. So deuteromycetes include disease-causing fungi and beneficial fungi. 2015 NEET had a matching question, which is correct. Alternaria, sexual reproduction absent, deuteromycetes. That's the correct match. The other options had wrong combinations. So remember, Alternaria is a deuteromycete, and the defining feature of deuteromycetes is absence of sexual reproduction. Moving to kingdom Plantae. This kingdom includes all eukaryotic chlorophyll-containing organisms commonly called plants. Pretty straightforward, right? But there are exceptions. A few members are partially heterotrophic, like insectivorous plants. Examples are bladderwort and Venus flytrap. These can photosynthesize, but they also capture and digest insects to get nitrogen and other nutrients. Then there are parasites like Cuscuta. This is daughter. It completely lacks chlorophyll and depends on a host plant for all its nutrition. So even though Plantae is defined by having chlorophyll, there are some members that break this rule. Plant cells have an eukaryotic structure with prominent chloroplasts. That's where photosynthesis happens. And cell wall, mainly made of cellulose. Remember, fungal cell walls have chitin, but plant cell walls have cellulose. The major groups included in Plantae are algae, bryophytes, mosses, pteridophytes, ferns, gymnosperms, conifers like pine trees, and angiosperms, flowering plants. You'll study these in detail in Chapter 3. Now here's something unique to plants. Their life cycle has two distinct phases that alternate with each other. There's a diploid, sporophytic generation and a haploid, gametophytic generation. This phenomenon is called alternation of generations. So unlike animals where the diploid stage dominates and the haploid stage is just the gametes, In plants, both stages can be substantial. The lengths of the haploid and diploid phases, and whether these phases are free-living or dependent on others, vary among different groups in plants. In mosses, the gametophyte is dominant. In ferns, both are independent. In seed plants, the sporophyte is dominant and the gametophyte is reduced. This is a fundamental concept in plant biology. Last kingdom, kingdom Animalia. This is characterized by heterotrophic eukaryotic organisms that are multicellular, and their cells lack cell walls. No cell walls is a key difference from plants and fungi. Animals directly or indirectly depend on plants for food. Even carnivores depend on plants indirectly through the herbivores they eat. The mode of nutrition is holozoic, by ingestion of food. Holozoic means they eat food, take it into their body, and digest it in an internal cavity. This is different from fungi, which absorb nutrients from outside. Animals store food reserves as glycogen or fat, not starch like plants. This is a biochemical difference. Animals follow a definite growth pattern and grow into adults that have a definite shape and size. Unlike plants, which have indeterminate growth and can keep growing, animals reach a certain size and stop. Higher forms of animals show elaborate sensory and neuromotor mechanisms. Complex nervous systems, specialized sense organs for vision, hearing, smell. Most animals are capable of locomotion. They can move from place to place. Sexual reproduction in animals is typically by copulation of male and female, followed by embryological development. The developing embryo goes through specific developmental stages. Salient features of various animal phyla will be described in Chapter 4, so we won't go into detail here. Now we come to some organisms that didn't fit into Whittaker's 5-kingdom classification: viruses, viroids, prion, and lichens. Let's start with viruses. All of us who have suffered the ill effects of common cold or flu know what effects viruses can have on us. Viruses did not find a place in classification because they are not considered truly living if we understand living as organisms that have cell structure. The viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Outside a host, they're basically inert crystals, lifeless. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. So the question is, would you call viruses living or non-living? Outside cells, they're non-living. Inside cells, they're active. It's a gray area. The word virus means venom or poisonous fluid. That gives you an idea of how people viewed these infectious agents when they were first discovered. The history of virus discovery is fascinating. Dmitry Ivanovsky in 1892 recognized certain microbes as the causal organism of the mosaic disease of tobacco. These were found to be smaller than bacteria because they passed through bacteria-proof filters. So they call them filterable agents. M. W. Bajerink in 1898 demonstrated that the extract of infected tobacco plants could cause infection in healthy plants. He named the new pathogen virus and called the fluid Contagium vivum fluidum, which means infectious living fluid in Latin. Then W. M. Stanley in 1935 showed that viruses could be crystallized, and these crystals consist largely of proteins. This was revolutionary. You could crystallize an infectious agent like it was a chemical, not a living organism. Viruses are obligate parasites. They absolutely must infect a host to reproduce. They're inert outside their specific host cell. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. Here's the key point. No virus contains both RNA and DNA. It's always one or the other. A virus is essentially a nucleoprotein, nucleic acid plus protein, and the genetic material is the infectious component. The protein coat protects the genetic material, but it's the DNA or RNA that actually carries the instructions for making new viruses. Let me show you a 2015 NEET question that tested this history. It asked, select the wrong statement. The answer was, the viroids were discovered by D.J. Ivanovsky. That's wrong. Ivanovsky discovered viruses, not viroids. The correct discoverer of viroids was T.O. Diener in 1971. The correct statements were: the term contagium vivum fluidum was coined by M. W. Bajerink. Mosaic disease and tobacco and AIDS are caused by viruses, and W. M. Stanley showed viruses could be crystallized. So make sure you know who discovered what. Ivanovsky, viruses. Bajerink, coined term and confirmed viruses. Stanley, crystallized viruses. Diener, discovered viroids. Now, let's talk about virus structure and types. In general, viruses that infect plants have single-stranded RNA. Viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. So animal viruses show more variety. Bacterial viruses or bacteriophages, viruses that infect bacteria, are usually double-stranded DNA viruses. So the type of genetic material often relates to what host the virus infects. The structure of a virus is relatively simple. There's a protein coat called capsid. This capsid is made of small subunits called capsomeres. The capsid protects the nucleic acid, the DNA or RNA, inside. These capsomeres are arranged in helical or polyhedral geometric forms. Helical means they spiral around like a helix. Polyhedral means they form a many-sided 3D shape, like a soccer ball. This arrangement of capsomeres is quite beautiful when you see it under an electron microscope. A 2014 NEET question asked, which virus shows coiled RNA strand and capsomeres? The answer is tobacco mosaic virus, TMV, not poliovirus, measles virus, or retrovirus. TMV has a helical arrangement. The RNA is coiled and the capsomeres spiral around it. This is a classic example used to teach virus structure. Another 2014 question asked, viruses have what? The answer is DNA enclosed in a protein coat. But note, it could be DNA or RNA, never both. The wrong options said prokaryotic nucleus. Viruses don't have any nucleus, single chromosome. They have genetic material, but it's not organized as a chromosome. Both DNA and RNA? Wrong. It's always one or the other. So, virus equals protein coat, plus either DNA or RNA. Viruses cause many diseases. In humans, mumps, smallpox, herpes, influenza, the flu, and AIDS is also caused by a virus, HIV. In plants, the symptoms can include mosaic formation, patchy discoloration of leaves, leaf rolling and curling, yellowing and vein clearing. The veins lose their green color, and dwarfing and stunted growth. The plant doesn't grow to normal size. These viral diseases cause enormous economic losses in agriculture. 2019 NEET asked, which statement is incorrect? The answer was, infective constituent in viruses is the protein coat. That's wrong. The infective constituent is the genetic material, DNA or RNA, not the protein coat. The protein coat protects the genetic material, but it's the nucleic acid that's actually infectious. The correct statements were, viroids lack a protein coat, viruses are obligate parasites, and prion consist of abnormally folded proteins. So make this distinction clear. Protein coat is protective, genetic material is infective. Now we come to even simpler infectious agents. In 1971, T.O. Diener discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be free RNA, just RNA, nothing else. It lacked the protein coat that is found in viruses. Because it's virus-like, but even simpler, they called it viroid. The RNA of the viroid was of low molecular weight. It's a small piece of RNA compared to viral genomes. So the progression is, virus has protein coat plus RNA or DNA, viroid has just RNA, no protein. NEET has asked about viroids multiple times. 2020 asked, which is correct about viroids? Answer, they have free RNA without protein coat. 2017 asked, viroids differ from viruses in having what? Answer, RNA molecules without protein coat. 2016 asked, which statement is wrong for viroids? Answer, their RNA is of high molecular weight. That's wrong, it's low molecular weight. So remember, viroids equal free RNA, no protein coat, Low molecular weight RNA, smaller than viruses, cause plant diseases. Now we go even simpler or stranger. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses but contained no nucleic acid, no DNA, no RNA. It's just protein. These agents are called prion. Prion stands for proteinaceous infectious particle. This was revolutionary because everyone thought you needed nucleic acid to transmit information. But prions show that an abnormally folded protein can cause other proteins to misfold too, spreading the disease. The most notable diseases caused by prion are bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle, and its analogous variant Creutzfeldt-Jakob disease, CJD, in humans. These are devastating neurological diseases where the brain literally becomes sponge-like with holes. There's no cure because the infectious agent is just a misfolded protein. These diseases showed us that infection can occur through mechanisms we never imagined. NEET loves testing prion. 2022 asked, mad cow disease in cattle and Creutzfeldt-Jakob disease in humans are due to infection by what? Answer, prion. Not virus, not bacterium, not viroid, prion. 2019 asked, mad cow disease in cattle is caused by an organism which has what? Answer, abnormally folded protein. So you see a clear progression in infectious agents. Bacteria have cells, viruses have protein coat plus nucleic acid, viroids have just RNA, prion have just protein. Each is simpler than the last. Last topic, lichens. Lichens are symbiotic associations. That means mutually useful associations between algae and fungi. The algal component is known as phycobiont. Phyco means algae, biont means organism. The fungal component is known as mycobiont. Myco means fungus. The phycobiont is autotrophic and the mycobiont is heterotrophic. So how does this partnership work? Algae prepare food for fungi through photosynthesis. They make organic molecules that the fungus can use. Fungi provide shelter for the algae and absorb mineral nutrients and water for their partner. It's a perfect division of labor. Algae are the food producers. Fungi are the miners and shelter providers. Both benefit from this relationship. The textbook makes this interesting point. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. They look like a single organism. The fungal and algal cells are so intimately associated that it's hard to separate them. This is one of the best examples of symbiosis in nature. Lichens are very good pollution indicators. They do not grow in polluted areas. Why? Because the symbiotic relationship is sensitive. If air pollution damages either the algae or the fungi, the whole lichen dies. So if you see lichens growing on trees and rocks in an area, that tells you the air quality is good. If there are no lichens, that's a warning sign of pollution. Scientists use lichen presence and diversity as biomonitors of air quality. 2019 NEET asked, which statement is correct about lichens? The correct answer was, lichens do not grow in polluted areas. The wrong option said, lichens are not good pollution indicators. False, they're good indicators. Algal component is called mycobiont. False, it's phycobiont. Fungal component is called phycobiont. False, it's mycobiont. So remember, phyco equals algae, myco equals fungus, and lichens equal pollution indicators that don't grow in polluted areas. This concludes our chapter on biological classification. Make sure you review the 5-kingdom system, understand the characteristics of each kingdom, know the examples, and remember how viruses, viroids, prion, and lichens fit into, or don't fit into, this classification scheme. Thank you for watching. Download the Iyal.App.
Meet Iyal, your AI study companion. Transform PDFs into smart study guides, practice tests, and flashcards for UPSC, NEET, banking, and any competitive exam.
Master your exam preparation with Iyal.